Good evening, sir and madam. As you all know, this is Science Dialogue series, free webinar series. Main purpose is to increase knowledge pool in bioscience stream or related stream which helps bioscience. In this series, we have invited very renowned scholar, Dr. Amrendra Ajay. He's basically from Bihar, did his BSc from BN College, MSc from IIT Mumbai or Bombay, and then PhD from NCCS Pune, and then postdoc. बहुत सारे BSc MSc स्टूडेंट को पता भी नहीं होगा. So he is postdoc from Harvard Medical School, that is top school in world research के मामले में, and he has published several research paper, review paper in top journal like Blood and Molecular Cancer and several good journals. And thank you, Amrendra Jaji. Your name is visible, Poonam Kumari. So for participants who are watching him, he is Amrendra Ji, Dr. Amrendra Jaji. Name showing Poonam Kumari, maybe her wife name. या बच्चे कौन है?
You're right. She is my wife, and she has this computer login,
yes.
so it took already.
Yes. So welcome, and we heartily, Hannah, thankful to you for accepting our invitation and ready to share your knowledge to our people. Our means this is open for all, but yes, those who are interested in learning science. Mostly they are from India, but few participants may be from outside India also. And participants are from UG to faculty, scientist, dean, director, all. This is open for all. So thank you again. Now you can start.
Thank you, Dr. Gopal, for giving me a good introduction. And first of all, I would like to thank you especially because I think this forum is very important to propagate the science across the border and also within the community. So I realize the value of these talks, and we should do more and more talk from a different people and different experts. And you are already doing it because I can see that tomorrow is another lecture scheduled. So I appreciate your efforts and your motivation for doing this. And I know it is an extra hour for everybody, and I appreciate that. Second of all, I would like to thank all the students who are here, spending their weekend with me. And my only purpose is that you can learn something good from my lecture, which you have never heard or never learned. Even a sentence adds to the ocean like a drop. This is how learning works. If I'm going too fast or too slow, you can tell me in between. Feel free to raise your hand. Also, I have made my presentation for 40 minutes and then 20 minutes of discussion because I want you to know me better than just talking. I like that format better. And
yes,
of course,
yes.
Dr. Gopal can moderate that as well.
Yes. This is interactive session. So yes, interaction is more important.
In between, also, if you think that I'm not getting this, just raise your hand, and Dr. Gopal will let me know that we have a question, and we can talk. I'm not in hurry, so just be sure that this is a weekend, so don't worry about another meeting I have after this. So I will try to answer all your questions that I know. With that, I would like to share my screen. I think you.
Yes. Full screen kar lijiye.
Okay, so today I'll be talking about role of microRNA in autoimmune diseases. I'll focus on one disease, which is called multiple sclerosis, but this is true for many other diseases, concept-wise. I have some disclosures that I have to disclose because we get financial conflicts when we don't disclose them. So these are my disclosures in terms of parents and founding a company and co-founding. a company. So without delay, I will explain you what are the microRNA. So if you look at a microRNA, of course this is RNA, and I assume like many of you know what is RNA. So if you look at this microRNA, most importantly, this is not functional. So this will not bind to the RNA molecule and to the regulation of the gene. What happens is this RNA, which is 20 to 26 nucleotide, and this is very regulated as other things. You have heard about proteins and all those things, how they are regulated. So there are dishers and rosa that cut them when they are needed by the cells. And this is small fragment, 20 to 26 nucleotides, are the active microRNA. And this is the concept is similar as siRNA. You have heard about short interfering RNA. These microRNA, they bind to the mRNA and they pause the transcription and translation, or they degrade it depending on how efficiently it binds. So what happens when the mature RNA, you can see here the mature RNA is uh, in the cell. If there is a perfect binding to the target mRNA, what they do is they cleave and degrade the mRNA, so there is no transcript available for translation. If and there is also imperfect binding, all the sequences are not exactly matching to the target mRNA. In that case, it binds, but it inhibits the translation of the protein. So what happens when there is no translation? There is no protein made, right? So even if you are looking at RNA assays where you look for RNA binding or cleavage, you might not see a change in the protein in the mRNA, but you may be able to see the change in the protein, because if the translation is inhibited, you will not see difference in the mRNA level. So, what is the autoimmune disease? Many of you have heard, but I just give you an overview of like very brief overview. Um, so these are on the slide. You can see 10 different autoimmune disease that comes from different organs. So I will focus to mainly on multiple sclerosis, which is basically a disease in the brain. I will tell you more about this disease, what is the pathophysiology of this disease. You must have heard about asthma, which is like coughing and all the difficulty in coughing. This is very common in our India. Celiac disease, which is a disease in the intestine. Eczema and psoriasis. You must have seen your family members or somebody having this eczema. My dad has a severe eczema. Rheumatoid arthritis, which is the pain in the joints, and this can happen whole body, but mainly in the knee, especially in the old ladies. We see more knee pain, and that is because of this autoimmune disease, autoimmunity. Systemic lupus erythematosus. So this actually affects whole whole body, and the symptoms are like a mix of all these asthma, celiac disease, and coughing. Hashimoto thyroiditis means that there is a problem with the thyroid, and that is also because of the autoimmune and activation. So why it is important and why it is autoimmune? Does any of you know? Because I want to know if you are following me right. If I don't see a hand, I'll
They are
move forward.
listening. They are listening.
Okay, good.
Yes.
So as I told you, I will give you a little more introduction about pathophysiology of multiple sclerosis. So what happens first? So this is a neuron. This is the body of the neuron. And these, I want you to focus on these cells. These are neuron, and these are myelin cells. These are myelinated cells. So what happens is when B cells and T cells, they find an antigen which is inside the body, they bind to these myelin positive cells. This is the first step that happens, and they start attacking these cells. And then after that, what happens is there's a lot of cytokines and chemokines that are released into the microenvironment, and these causes activation of other immune cells and T cells to come. And after that, these antibody basically bind and that attack. Sorry, and they release antibodies that attacks myelin cells, and they basically inhibit the function of myelins and degrades the myelinated cells, and finally leads to injury to the brain. And this is a injury that many of you might know that neurons are very difficult to regenerate. Like other cells, like liver cells, if one cells die, other cells pop up and they repair it. Kidney cells, heart cells, they repair very well. But brain cells, they are limited from the birth and they don't repair well. So people are working on how to regenerate these cells so that they can repair the brain. But this is one of the example how these cells die and they lead to uh, disease. So is it important? Is these cells, T cells, B cells are important? Yes. This year you have heard that the T cell uh, biology got Nobel Prize in 2025. So Mary, Fred, and Simon, these three people, what they found is the exact discovery that we are kind of talking based on those basic findings. So they found in 1980 that how so T cells are can attack our cells, right? But they are not attacking our cells, right? They are attacking foreign particles, viruses, bacteria that come in our body. So he found what he did is he thought these cells are good for preventing us from disease or bacteria pathogens. So Simon, what he did is in 1980 and later, 10 years later, Fred and Mary did similar experiments. What Simon did is he made a mouse. He made a mouse without the thymus. So the T cells were not produced in these mice. And he was thinking that these mice will have a lot of infection and they will die because of they don't have a capacity to fight with the antigens or external stimuli. But the results were very shocking because what he found is these mice were having a lot of problems in the development of many organs and they died prematurely. So that was very concerning to him. And at that time, think about 1980 when I was born, the science was still primitive. So what he thought is, what is happening with these mice? Why they are dying? Because in the body of the mother's womb, there were no foreign particles coming. So because these mice, as you many of you know, we grow them in pathogen-free status. Like there is no, they get like filtered air, like a mask, but better than that, and they get food and everything sterilized. Their housing is very well sterilized. So there's no pathogen coming to the mice. This was surprising to them, and they took some time to figure out what it is doing. And later, this is a recent review, 23. He found that there is a transcription factor called FOXP3. I want to show you here in the second column, which basically inhibits the activation of T cells. And if you imagine in the third column, you can see if there is no FOXP3, there will be no suppression and there will be a disease, autoimmune disease called IPEX. But if you have a FOXP3, it will suppress and there will be no immune response to the self. Means the self cells will be, our body cells will not be attacked. So this was such a big discovery that time that nobody thought this is this is small. And then people were thinking of, there are so many papers and so many disease that has been studied in the context of autoimmune disease based on FOXP3 and other transcription factors. So basically, uh, this was a discovery for the regulatory T cells. And you, as you must know, they are Th1, Th2, they are T17, Th17, and other kind of cells that you will see later. So what we do in the lab in terms of autoimmune disease, and these are all our data that we do in the lab. So we are studying the role of microRNA 92A, and we have other products going on related to microRNA. But today I will discuss one part of microRNA, which is microRNA 92A in MS, multiple sclerosis. So I will give you some example and some data to convince you that these microRNA are important for the development of multiple sclerosis. So to start with, what we did is we how do you study microRNA or any disease in the mice? So for the mice, we need experimental model, right? So suppose you are studying cancer, so you generate you inject the cells in the cancer in the mice, and then you grow them, and then you treat with the anti-cancer drug. Similar to that, what we did is we have a mouse model where it is called EAE, which is experimental autoimmune encephalitis. So what we do is in EAE, we put a peptide in the mice. What it does is it opens the blood-brain barrier. So T cells and all those, any cells cannot go to the brain. So how it goes in human also. So the blood-brain barrier is ruptured because of the disease, and then the cells go to the brain. That's exactly what we do with injecting a peptide called MOG, which is a small peptide, 15 kilodalton. It breaks the blood-brain barrier, and then cells can go inside the brain. And after that, what happens is when the cells go in the brain, it basically, I told you in the previous slide, it happens something like this. The mouse brain. These cells attack, and then they finally cause the injury to the brain cells. And we scored these based on how the mice is doing. How do you know? Because if you leave the mice walking, if they have a paralysis, because this causes paralysis to the mouse, so mice will not be able to walk properly. You can score them based on 1, 2, 3, 4. 1 being the lowest score, or 0 being the no disease, 1 being the lowest score, and 2 being the highest, and 5 being the most.
स्क्रीन शेयरिंग वो स्टॉप हो गया, प्लीज एक बार वो कर दीजिए स्क्रीन शेयरिंग, कुछ सेटिंग में प्रॉब्लम आ गई, जस्ट अगेन.
Okay, I will do it right away. अभी बंद हुआ. Oh, that's not good.
Yes.
Okay, can you see this now?
Yes.
Okay. So as I was telling you, EAE means if you have we give a clinical score to the mice based on how much injury is there, EAE there. So 0 is no disease. 1, 2, 3, 4, 5. 5 is the maximum score, means very severe disease. And so what we looked at is in EAE mice, naive mice is the control mice without injury, any EAE, and control, and EAE is the mice with the disease. So we isolated the spleen and central nervous system, figure A. And we looked at the microRNA 92A. And you can see clearly that in the disease mice, there was increased yeah, microRNA 920A, 92A. This is telling you that during the disease, these microRNA is upregulated. So that gives us a clue that there's a change in the microRNA, which may play some role during the disease. Then we developed a microRNA 92A knockout mice. So basically, you delete that microRNA 92A from the genome, and then you do the EAE. So black dots are wild type mice without any manipulation of the disease of the microRNA, and red bars are basically microRNA 92 knockout mice. So you can clearly see that after 15 days, these experiments are done for a month. So after 15 days, you see that there's a decrease in the clinical score of these microRNA 92A knockout mice. So this shows us that microRNA 92 is basically causing the disease, and deletion of this microRNA is protective against the disease. Are you able to see my next slide?
Yes, it's visible.
Okay. So then we looked at, so these are same data that I'm showing you here. I will move forward. Then what we did is we looked at the T cells in the brain. So uh, we we did develop the disease. We induced the disease using the same EAE model, and we looked at the CD4 positive cells, which is a T cell marker, using a FACS analysis. So you smash the brain, and then you do the FACS with the CD4 positive staining, and you can see that in these mice, all these markers of T cells and other immune cells like interferon gamma, IL-1, IL-17A, GM-CSF, and other were decreased as compared to the wild type mice. So what this tells us is there's a reduced number of cells, and there's a reduced cells with inflammation in these mice in the brain of these cells. Okay. So next, we look at the regulatory T cells, and as I told you, the discovery of FOXP3, these papers were published like 2 years before. So this FOXP3 was very well studied, and everybody want to see what is happening to the regulatory cells in these mice. So we looked at the FOXP3 positive cells, T cells, and you can see that this wild type has 8% of the T cells, which is positive for FOXP3, but the microRNA 92A knockout mice has 15%, almost doubled the amount of FOXP3 positive cells. And these are all in diseased mice, wild type versus knockout, I'm showing you. So you can see that FOXP3 in the T cells, FOXP3 population was increased in the microRNA 92A's knockout mice as compared to the wild type mice. This is the quantitation. So what these data suggest to us that microRNA 92 knockout mice have less T cells, but they are more kind of regulatory T cells. So one of the elegant experiment that we did is we looked at the function of what is the exact function of T cells. Does it matter in these mice? So what we did is we did adaptive transfer of T cells. So these are called, these are very uh, crucial experiments done by, established by many researchers in T cell biology. So what you do is you have the wild type mice to irradiate them with the UV so that there is no immune cells, because immune cells have a half-life and they keep coming, growing. So when you irradiate them, the bone marrow cells are dead. So there is no immune cells in the circulation. And then you can isolate the T cells from wild type and microRNA knockout mice and transfer them to the RAG deficient mice, which does not have T cells, irradiated ones. And then you induce the EAE in these mice. And I'm showing you the result on the right in I, the graph. So what you can see is microRNA 2192 in knockout mice, they develop less EAE or less MS symptoms as compared to the wild type. You can see the black dots. So that difference was very promising, and we were very happy to see that even the whole mouse is getting the same kind of injury, but the T cells are the important contributor to the MS in these mice. And the microRNA 92 knockout mice, they don't develop uh, severe EAE in these mice. So confirming the role of microRNA 92 in EAE in a different setting. Then we look for how this microRNA 92 is uh, protecting, knockout is protecting the mice, because it is important to know in terms of therapeutics. And I want to emphasize in general to all the students that once you have a protein or a gene or mRNA or a microRNA increased or decreased in the disease, that tells you that there is a role that it may play, but you have to find a mechanism how it is doing it, because the the target itself may not be druggable, because it may be so crucial molecule that if you inhibit it or if you overexpress it, there may be so many side effects. So if you know the mechanism of that target, you may design a drug against it. And I want to also tell you in terms of pharmacology and pharmaceuticals point of view, that many drugs that has been developed has a target which is very well studied in the field. So if the target is not well studied, there is a chance that it will fail in the clinic or before preclinical studies, because this may have a lot of side effect or this may not work uh, per se. So in a nutshell, we are trying to look at the how microRNA 92 inhibits Treg differentiation. So we found we did a RNA-seq, and I'm showing you a small plot here where we found that these are the genes FOXO1, STAT5, and others. They were basically increased in the microRNA 92 knockout. So yellow is increased and white, blue is decreased. So you can see on the top FOXO1. So we thought that okay, microRNA 21. So what happens if microRNA 21 knockout is there? If FOXO1 is increased, that means microRNA 92A was inhibiting FOXO1 expression, right, in wild type mice, and that's why it is increased when you knock it down, because the expression goes up. So we looked at the FOXO1, which is our top candidate, and we looked at the sequence match. So you can see this is microRNA 92A in mice, and later I will show you similar data in human. This is mouse FOXO1 3' UTR, where microRNA binds. And we were able to find that these are the regions where there was a match between these two. And we then designed a construct, luciferase construct. So what happens is we clone these FOXO1 3' UTR in the luciferase construct, where you have the, after this mouse FOXO1 UTR, you have the luciferase gene. So what does this mean? That if something binds to this FOXO1 3' UTR, there will be transcription of the luciferase gene. And we read the luciferase gene using a machine. It's basically, luciferase is firefly, right? So they glow and you read the glow. More the glow, more the luciferase activity. So these are the empty vectors, three, and these three are the vectors with FOXO1 3' UTR. And you can see first the orange one in the middle is microRNA 92A mimic, means you are overexpressing the microRNA in these cells. And this is microRNA 92A inhibitor, where you are inhibit using a small uh, nucleotide to inhibit this microRNA 92. And the results were very good because you can see that when you overexpress microRNA, there's a decrease in the luciferase activity, and there was an increase after inhibitor treatment. So this data tells us that microRNA 92A targets FOXO1 for its transcription. Next, what we did, the facts analysis showing you that we have like 32% of the T cells which are positive for FOXP3, which in the controls. In the knockout, it was more, as I showed you before. There were like it is a good target because when you knock out FOXO1, you see that there is restoration of FOX3 positive cells. And this is the quantitation for the same, that there was an increase in the FOX in the microRNA 92A mice with control siRNA, which was decreased by or bring back normal by the FOXO1 siRNA. So next we question whether we can target these microRNA and see if we can inhibit the EAE in these mice. So what we did is we injected microRNA inhibitors. So as I told you, these are 15 more nucleotides that is complementary to microRNA 92A, and they bind to the microRNA 92 and inhibits the binding to the mRNA. So what is happening here is you can see that this is the clinical score, EAE score. So control inhibitor, we injected with control inhibitor, we don't see much change in the score, but our microRNA 92 inhibitor decreased the score. This means these mice were protected from EAE. What does this mean? This means that you can target microRNA 92A and this will protect mice from EAE. This was very, very encouraging data for us because, as many of you know, siRNA is already in the clinic, but microRNA is not in the clinic yet. So people are trying to design inhibitors, and we got these inhibitors from a company, and we designed these to use it in the mouse. And many times you can imagine many of these biologicals, natural occurring biologicals, they don't work well. You know why? Because they may not be stable inside the body. But this was encouraging for us for the first time. So we were able to target microRNA 92A, and this was showing us a phenotype that we can protect these mice from EAE. Next, what we did is we looked at the FOXP3, means how microRNA inhibitor is working. So you can see this is control inhibitor, and this is microRNA 92A inhibitor. So you can see there's an increase in FOXP3 positive cells. These are all T cells that were isolated. So you can see there's almost 2.5-fold increase in the FOXP3 positive cells, and this is the quantitation showing you that after microRNA 92A inhibitor treatment, we see an increase in FOXP3 positive cells, T cells. So then next question we ask is it relevant to the MS patients? Means the patient, because many times we do a nice cell research, nice mouse research, but that does not translate to the human. So, because there's a gap between human, mouse, and cells, as you many of you know, it's not the same system. So, what we did is we got these T cells from the patients, and we did some study here. We used the microRNA 92 inhibitor in the T cells that we isolated from patients, and you can see that after inhibitor treatment, we saw that there's a like CD4 T cells were decreased. microRNA 92A expression was decreased in the inhibitor treated cells. So this is telling us that, okay, our inhibitor is working in the human. And this is a different inhibitor than the mouse, because human and mouse, they have different seed sequences. So next, what we did is we looked at the FOXP3 positive cells, and you can see this is control, and this is the 92 inhibitor. You can see there's a twofold increase in the FOXP3 positive T cells, which is telling us this is the quantitation of the same. So these data are telling us that if you inhibit microRNA 92A in these T cells, human T cells, you will see an increase in FOXP3 positive cells, and these T cells are regulatory T cells. With that, I would like to thank you all for listening to me. And
Thank you. बहुत अच्छा.
I would like to acknowledge the funding from different sources and staff and scientists in the lab that we are involved in. And I will be happy to take any questions or anything that you think you did not get.
Yes. Now platform is open for discussion. But before asking question, you must introduce yourself and then ask question.
Ashish Vyas, actually, I have a question related to how you selected this microRNA on the basis of what?
Oh, Ashish, this is a very good question. So in 2018, we did a RNA-seq analysis that was more like exploratory from the patients who has MS. So we are associated with the MS Center at Harvard Medical School. They have the patient samples, and we isolated RNA from them, and then we did the RNA-seq, and we looked at the many microRNA, we looked at many mRNA, and also other kind of RNAs. But for the microRNA, we found that many of the RNAs were upregulated in the MS patients, but this was one of them, the top hit. And we are going through the list, how many we can study. Good question.
Ashish sir, please introduce. Ashish sir is also our speaker. Ashish sir.
Uh, Thank you so much. Yeah. So I'm
आपका
working here.
कैमरा भी नहीं ऑन है. Please introduce कर दीजिए sir.
Hello. Yeah, so actually I'm working as a faculty at Amrita Research Center in area of liver immunology and looking for pathogenesis in acute on chronic liver failure cirrhotic patient. So we're also looking for microRNA carried by the exosome and looking for early biomarker detection for liver advanced injuries. Yeah. Thank you so much.
Oh, that's very
Thank you.
nice. Because we
Other
have
participants? Yes.
Yeah.
Please continue sir.
Yeah, we have another project going on in the lab where we are looking for exosome-mediated microRNA discovery, biomarker discovery in urine, mainly for kidney disease.
Okay.
I have shared feedback Google form in this chat box. Those who need e-certificate, please fill that feedback form now. Okay, any other participants have any question, any suggestion, please unmute and speak. Other participants, कुछ questions नहीं है? Any question? Somelia Mishra, you are here.
Hello,
Yes,
Doctor sir.
हाँ कुछ questions है?
No, sir. currently no, अभी नहीं है.
इंट्रोड्यूस करो आप.
Okay, sir. I am Soumya Mishra. I am currently working as a junior research fellow or a PhD student at Baba Atomic Research Center. So, yeah, currently I am working in ACTREC also, and my research area is radiation biology. And I listened to your lecture, and it was very intriguing for me. Thank you, sir. If any questions will be there further, I will mail you, sir.
Good.
Yeah, you can email me. I think Dr. Gopal had my email. So if you want to email me, email me. Just email me.
Any other participants have any question? हार्वर्ड में जाना कैसे वही पूछ लो, कुछ पूछो.
Hello sir. Hello sir. I'm Nikhil Kumar Sharma from Bihar.
हाँ बोलिए निखिल.
I am pursuing my stem cell and regenerative medicine masters in stem cell in DY Patil Medical College, and I want to work on exosomes also.
Oh, nice. So you are looking for exosomes from the stem cells?
Yes sir.
Okay, interesting.
For biomolecular uses and also for drug delivery.
Okay, yeah, exosomes are a good candidate for drug delivery.
निखिल शर्मा आपका PhD हो गया?
No sir, I am pursuing my masters.
अच्छा मास्टर्स कर रहे हो. Okay.
Yes sir. So I confused about from where I start and how I start about it.
So starting a PhD, so you are in second year or first year of master?
I'm in first year. I'm in first year right now.
So what do you want to study in for PhD? Do you have selected that already, or what do you think?
No. I just pursuing my stem cell. Then after I do in stem cells also, stem cell or exosome field.
Okay. So we have a faculty here and at Harvard. They work on stem cells, not in exosomes, but in general stem cells.
Yes, sir.
I'm also part of Harvard Stem Cell Institute.
तो क्या PhD में होगा क्या एडमिशन वहाँ?
PhD will be difficult. PhD will be difficult in Harvard. I'm telling you.
Postdoc जाए तो अच्छा है. PhD में तो वहाँ.
Postdoc is better, but you can try other institutes.
Yes,
because Harvard is a little difficult for PhD because we have a centralized system, and they look at many things. You know, the
यहाँ PhD करो, है ना?
Yes, sir.
इंडिया में बाद में पोस्ट डॉक में जाइए, उनके बाद तो हेल्प करेंगे आपको. अमरेंद्र सर भी हेल्प करेंगे पोस्ट डॉक में. PhD अच्छा जगह से कर लो और उसके लिए
you can try Europe. You can also try Europe for PhD.
Yes, sir.
There is a benefit because you can do PhD in 3 years.
Yes, sir.
So
institute से 5-6 साल लगेगा.
Yes,
US takes like 5 to 6 years. India takes 5 to 6 years.
Yes.
So,
yes.
And in Europe, it is easier right now because they are attracting more people because of the visa and tariff. So you might be able to get there. Second thing you can try is try like Europe. I think I like better. And
Yes,
the thing
how
is
I
also,
stay in touch with you?
Yeah. So my email is with Dr. Gopal, or you can write
email
it right now.
दूंगा. पहले तो आप MSc करो, email दे दूंगा, and I will connect all the participants or attendees here on LinkedIn. LinkedIn पर इनको tag करूंगा, and you all can connect with him.
Yes,
yeah. You can send me a request. It's Amrendra Ajay. I think my name is Poonam Kumari right now because my wife's computer.
But I will tag you with this photo. आप वो stop कर लीजिए presentation, तो आप पूरा वो देख भी पाएंगे सबको, participants को. Yes. So, any other participants have any question?
Thank you, sir. Thank you, sir.
Welcome. I think somebody has their hand raised.
Sir, can I say something?
Yes, bolo.
Sir, you might be aware of that Mr. Trump has cut the federal funding.
Yes.
Sir, what effect it has been playing on the role that on research and for us like who travel from India to abroad for our postdocs or PhD?
Yeah, so research funding is a major issue, and it's a driving force for any research, as you know. So there is a less funding for now, and for postdoc it does not matter too much because If somebody is giving you a position, that means they have the fund. So as per our rule, if I have funding for 3 years, then only I can offer you a postdoc position. So you might see that you are not getting a position, which is okay. But if you are getting a position, and if they have funds for 3 years, you can do a good research in 3 years. Because what NIS does is they give you funding for 5 years, 3 years, 4 years, and they allocate money for you. If I have a funding, I'm I can offer you 3 year postdoc. You are good for 3 years.
Okay,
But
sir.
for if I don't have funding, if you write to me, I might not give you the position. So that is one thing. It will be reduced. Number of postdocs will be reduced. But I think NIH is trying to do their best to start everything. But due to the government, we have to follow their regulations.
Shravani has raised hand. Shravani, question पूछना है और by mistake you have raised hand. Shravani. Okay. इसका कुछ unmute and speak, Shravani, if you have any question. हाँ रघु जी. Raghu,
Hello, sir.
yes, welcome. पूछिए.
How are you, sir?
I'm good. How are you, Raghu?
Sir, nice. It is fantastic presentation, sir. Sir, I am a postdoc fellow, currently working at IIT Gandhinagar with Professor Dinesh Bhatia. So, sir, I'm working on the we have
Very
like
well.
synthesized, characterized, and we have also did some in vitro biology studies. And
Okay.
now I'm looking for the in vivo model for the glioblastoma study. So you mentioned in your presentation that your work is mostly on the neurological disorders. So is there any chance to collaborate with your team? Can we do some collaborate work and complete our project in those studies?
Yeah, so that's a good question. We are always We always try to collaborate with people with similar expertise or the complementary expertise. So we don't do any glioblastoma, but I can connect with you somebody who is doing glioblastoma
Okay, sir.
because this is a completely different setup, like injecting the
Yeah.
tumor in the
Yes.
brain. I know somebody who does that, and I will talk to them. They're in the same division. I'll talk to them and I will let you know. So shoot me an email and I will go from
Sure,
there.
sure. I will
Amrendra
mail you,
ji,
sir.
Dr. Raghu has also delivered lecture. Generally, Indian
Okay, nice.
postdoc, they are very hesitant to deliver online lecture. He has delivered.
No,
And
we
his
should
boss has
promote that.
also delivered. His boss has also delivered 2 lecture.
Oh,
Dr.
good.
Dheeraj Bhatia. So,
No,
मतलब
Dr.
hard
Gopal, I
working का
think
मैं तो guarantee.
we should encourage PhD students and postdocs to talk because that is one thing that we are missing. And it was happening to me also when I came here. I was hesitant to talking the science.
Yes.
We should make that culture very strong. This is your daily life. You are doing science for like 5 years as a PhD. You should be able to speak even you are sleeping. And that's what I heard
वही
from
मैं ये
you.
प्लेटफॉर्म कर रहा हूँ. ज्यादातर जो पोस्टडॉक इंडियन है, बहुत सारे इंडियन पोस्टडॉक है, बट दे आर हेजिटेंट टू टेक, है ना. 50 पेपर है,
Yeah.
बट दे डोंट हैव 1 hour लेक्चर कंटेंट टू डिलीवर.
That is true.
व्हाट आई हैव ऑब्जर्व. तो रघु जी को बोला, इनके गाइड ने इंट्रोड्यूस किया. इन्हीं के फिर दूसरे कलीग है, मैं नाम नहीं लूंगा, बट स्टिल सिंस 1 year इज नॉट, है ना, मेकिंग कि भाई ले पाएंगे ऑनलाइन या नहीं ले पाएंगे.
Yeah. It's like a good thing. I think it's communication is very important.
इसमें सबका पोस्टडॉक का भी फायदा है और जो सुन रहे हैं उनका भी फायदा है कि नॉलेज पुल इंक्रीज होगा, है ना.
Right.
सो रघु इज हार्ड वर्किंग. मेरे तरफ से अगर रिकमेंडेशन होगा सो आई विल डेफिनेटली रिकमेंड, और हम आपसे रिक्वेस्ट करेंगे कि यू हेल्प हिम व्हाटएवर यू कैन.
Of course. I told you, like, definitely happy to help.
I will write mail you, sir. Okay.
Okay.
हाँ, मेल भी भेजिएगा और आपको व्हाट्सएप नंबर भी रघु देंगे, है ना. आप कनेक्ट में रहिए, व्हाटएवर ही कैन, है ना, करेंगे हेल्प. एनी अदर पार्टिसिपेंट्स हैव एनी क्वेश्चन?
Thank you so much.
Sir, good evening. I'm Shravani and I'm in my master's first year right now. And I'm also planning to like pursue my future in stem cell and regenerative medicines.
Mhm.
But I need some guidance. Like there are so many fields of research in this field. Like now you told us about microRNA, then there is CAR T therapy, gene therapies, and then there are MSCs. So I am like really confused. Like what all fields are related to what things? I just needed some guidance about that.
Yeah, so
श्रवणी,
definitely let.
उनको उनके सजेशंस पहले मैं बताऊं आपको, ये सारे टॉपिक जो आप बोले हैं ना, सारे टॉपिक पर लेक्चर है हमारे साइंस डायलॉग सीरीज में, उसमें इसमें यूट्यूब में. जस्ट गो थ्रू इट एंड देन आपका इंक्लिनेशन किधर है, आपका इंटरेस्ट किधर है, पहला वो आप देखो. क्योंकि आप सिर्फ टर्मिनोलॉजी सुने हो, है ना, ये है जीन थेरापी, ये जस्ट गो थ्रू दैट. 1 आवर लेक्चर्स हैं हर पर में, एंड देन यू कैन कनेक्ट. अब आप कुछ एडिशन बताएंगे तो बता दीजिए.
Okay.
Yeah, that's a good question. I think we were also confused when I was doing masters. So it's a very common question that we see, because you see a lot of terminology and you see new terminology, new field coming up. So I think in general, I will tell you the US perspective, and like despite of these funding issues in US, like NIH cut the funding and all those things. One thing that the administration recognized is the stem cell. So they have made a new institution called the Stem Cell Organoid Management Institute,
माइक बंद
SOM.
करो श्रवणी, म्यूट योर माइक. हाँ हाँ, बोलिए प्लीज, कंटिन्यू सर.
So the organoid is basically, we are also doing in a different project, like trying to model the fibrosis from stem cells. So we are making organoids from the stem cells, and those are the small tiny structures. This is a 3D structure like a ball. You can see different parts of the organ inside the body, inside the organoids. So there is a push, and this will be for all the diseases, probably brain, lung, heart, kidney. You try to find your choice, what you want to do, and then look for that, chase that basically. Like if you are interested in kidney, it's a personal preference. You might be interested in brain, so look for brain organoid where we can develop, and there are labs that are doing it. So it's a good good problem to have, but you can carve out your niche, what you like, and definitely I will be happy to discuss if you want. As Dr. Gopal suggested, first look at what you are looking for. If you can get this information from YouTube or any other place, or the lecture series.
हमने ट्रेंडिंग टॉपिक जो बायोटेक में है ना, अमरेंद्र जी, क्रिस्पर हो, जीन थेरापी हो, स्टेम सेल थेरापी हो, इस सब पर का 10-12 ट्यूब का एक हमने कोर्स टाइप का फ्री वाला एक साथ करके प्लेलिस्ट में कर दिया, ट्रेंडिंग टॉपिक्स ऑफ बायोटेक्नोलॉजी. बट स्टूडेंट दे आर थिंकिंग कि भाई उसको एक साथ सारा चीज, इंक्लिनेशन तो उसको दिखाना पड़ेगा वॉच करके कि किसका क्या स्ट्रीम है ना. 1 आवर्स लेक्चर जो है ना, बहुत अच्छा सा सिंथेटिक बायोलॉजी पर, टिश्यू इंजीनियरिंग का, सबका एक ही में डाल के ट्रेंडिंग टॉपिक का हमने एक बनाया है ना. बट दे आर नॉट वॉचिंग. अगर दे भी दिया तो उनको बहुत भारी लगता है.
नहीं, स्पेंड टाइम लगेगा ट्रैवलिंग. जब ट्रैवल कर रहे हैं तो मोबाइल पे भी आ गया यूट्यूब, यू कैन वॉच एनी टाइम.
Yes, sir. I will actually.
Yes.
Actually, I am not their student. I did not know about any of this. I got the link from my friend, and I got the recommendation also from a friend. So I am just getting familiar with all this. So I'll definitely go through the YouTube.
Yes, so I have given all the links. Our YouTube का link तो दे ही दिया है. We have multiple lectures from different experts from all over world. हार्वर्ड से, स्टैनफोर्ड से, है ना, मैक्स प्लैंक से, इंडिया में IIT से, JNU से, more than 150 experts have delivered lecture on different topics. उसके अलावा हमारा तो है ही, हमारे स्टूडेंट का है ही, समझ लो आप. So you can watch there are so many topics, so many things are there. So whenever you get time, you watch, and then interest आपको जगाना पड़ेगा. उस सारे में से कौन सा इंटरेस्टिंग आपको लगा है, then you can deep down to that. But basic understanding के लिए 1 घंटे का लेक्चर है.
Okay, sir. Thank you so much.
Yes, so subscribe that channel also. Joining वाला paid वाला नहीं, this is free, है ना. You must watch. Any other participants have any question? रोहित सालूजा जी को देख रहा था मैं अभी. रोहित जी हैं और he left. रोहित सालूजा जी, yes, welcome sir जी. Any question, any suggestion?
अभी मैंने देखा आई थिंक दैट इज अ गुड प्लेटफॉर्म, तो स्टूडेंट्स के लिए काफी हेल्पफुल रहेगा, और मे बी यू कैन टेक द क्वेरी फ्रॉम द स्टूडेंट्स कि उनको एग्जैक्टली क्या चाहिए, अकॉर्डिंगली यू कैन प्लान कि जैसे अगर बायोइन्फॉर्मेटिक्स में कुछ लेक्चर चाहिए तो वैसा कर सकते हैं, या उनको आगे एमएससी करने के बाद क्या क्या प्रोस्पेक्टिव्स हैं इंडिया में या अब्रॉड में, तो दैट्स अनदर थिंग अबाउट इट.
हर इंस्टीट्यूट के जो फैकल्टी हैं, आप समझो अच्छे इंस्टीट्यूट का हमने लेक्चर उसमें करके अलग अलग टॉपिक पर मैं डाल दिया यूट्यूब में, और 1 आवर. जैसे जनरल यूट्यूब में क्या होता है 5 मिनट का लोग डालते हैं, उसमें कहानी पता नहीं चलता है. अब CRISPR पर 2 घंटे का, जीन थेरापी पर 2 घंटे का, आर यू गेटिंग, मींस कंप्लीट एक आइडिया लोगों का आ जाएगा उसमें. कुछ बाद में हुआ कि कुछ बच्चे बोले सर हिंदी में हो, तो हमने जीन 11 का एक कमेंट्री टाइप का बना करके 30 चैप्टर्स का दे दिया, ताकि बेसिक अंडरस्टैंडिंग नॉन साइंस वाले को भी या बेसिक जो बीएससी वाले स्टूडेंट है उसको भी पता चले. ऑल 30 चैप्टर्स हैव बीन कन्वर्टेड एज ए कमेंट्री एंड वी हैव एडेड. बट पढ़ना तो पड़ेगा स्टूडेंट को.
Right. Now, this is a good resource that Dr. Gopal is saying. This is a good resource. You go through it. You may not like many topics, but you like whatever you like, and you go deep dive into that. This is how it works. Like even for the research, when you are doing a PhD, you will not know anything about the topic, but you will look for the literature in the PubMed. Then you will identify what is relevant to me, and then you make a project.
हमने आसान कर दिया कि पेपर नहीं पढ़ना पड़ेगा, नहीं तो डाउनलोड करके पेपर पढ़ो, उसको फिर उससे निकालो. 1 आवर्स में कंप्लीट स्टोरी जो है ना, मतलब इस टाइप का हमने किया है.
और ये बहुत जरूरी है, because whoever is talking, they have done all the homework for you, so you are getting the gist of it, like summary of it, what is in the field. That's the
अब
good.
तो ऐसा हुआ है कि किसी को कॉन्फ्रेंस कराना रहता है तो बोलते हैं कि अच्छा स्पीकर हमको दीजिए. बोलता हूँ क्या थीम है, उसमें से वो जो लेक्चर हुआ है उनको वो लिंक भेज देता हूँ कि देख लो उसमें से कौन सा स्पीकर आपको अच्छा लग रहा है ना, एंड देन दे कैन सेलेक्ट. फिर हमको फोन नंबर चाहिए तो मैं दे दूंगा, पहले देख लो आप, फिर फोन नंबर चाहिए स्पीकर के लिए तो कर लो. या किसी को कोलैबोरेटर चाहिए उस टॉपिक पर, तो मैं बोलता हूँ भाई वो है इस टॉपिक पर, मान लो microRNA है तो भाई अमरेंद्र जी हैं या फलाना है, आप देखो यू कैन कनेक्ट विद देम.
राइट.
क्योंकि जनरल यूट्यूब चैनल में क्या होता है, एक ही व्यक्ति अपनी आइडिया पूरा डालते हैं ना, छोटा छोटा वीडियो करके नंबर बढ़ाते हैं. हियर वी हैव इनवाइटेड सो मेनी, एंड आई एम लकी कि पीपल आर रेडी टू डिलीवर, है ना. बहुत सारे
लोग will help you. If there is a good cause, people will help you for sure.
yes. So thank you very much.
Okay. Thank you
for
Dr. Gopal. Thank you all the students.
lecture. बहुत अच्छा रहा. Still, if any participants have any question in future also, you can connect with me. I will connect you with today's speaker and other speaker also. हम हमेशा ही बोलते हैं आप अगर inclined हो आप पूछ सकते हो अपने, है ना, कि कौन से साइंटिस्ट किस टॉपिक पर कर रहे हैं, क्या कर रहे हैं. हम लोगों को पीएचडी टाइम में तो पता ही नहीं चलता था, है ना. या तो आप गूगल से सर्च करो, फिर उनका पेपर पढ़ो, फिर पेपर में पता चला कि उनका जो स्टूडेंट है दे आर मोर एक्सपर्ट देन देयर गाइड. Sometimes. So technical अगर हैंडल हो रहा है तो they are not able to help. अभी तो मैं बोलता हूँ कि आपको कहीं प्रॉब्लम आ रहा है बायोलॉजी में एक्सपेरिमेंटली, so I will connect you with that expert.
That is true. Yes.
So thank you very much. Keep in touch.
Thank you. So bye everybody.
Thank you very much everybody. Please send feedback. Feedback link गणेश जी चैट बॉक्स में फीडबैक लिंक हमने ऐड कर दिया है. If you need e-certificate, you can fill. If don't need, no need to fill. ये हमने chat box में डाला है. And good night to all. Keep in touch. I request again you all to join our social sites so that you will not miss. And keep suggesting how to make it better. How to make it more, you can say, better and more penetrable, so that that knowledge go in this society. ठीक है. Good night.